Welcome our first speaker for this session, Thulani Makhalanyane, who will be speaking to us on the role of microbial communities in advancing One Health.
So, good afternoon, everyone. Good afternoon, everyone. Thank you. About a couple of weeks ago, I was talking with Matthew Hutter, who is the journalist who kind of wrote all the profiles. And as we were discussing, I was telling him a little bit about my work and what I do. He suddenly just looked at me and said, "And you are speaking after lunch." Good luck with that. So we'll see how this goes. So I'll be talking about what we can do to advance One Health through the African Microbiome Project. So maybe before I get started, I would like to thank the foundation for providing the logistics and inviting me to come and give this talk. I'd also like to thank our sponsors who've supported the research that we do, who've supported student bursaries and the very costly genomics research that our lab works on. So if your logo is not here and you are keen to sponsor us, please call me. So this might be a slightly controversial start, but just remember, I'm not the one saying this, but it's the authors of this paper, who wrote a paper a few years ago, probably 10 years ago, saying that biodiversity is so much more than legs and leaves. They start this paper with a very controversial opening sentence, saying that despite suggestions from popular science programs, planet Earth is not owned by insects or humans. It's a microbial world. And then they end off the paper by saying that it is time to acknowledge the presence of and study microorganisms beyond those that generate tourist revenue. So I'll try through my talk and maybe give you a little bit more context in terms of our thinking and why we think microbes are actually at the heart of the One Health concept. So microorganisms have been here since the dawn of time, right? It was the first oxygenic microbe that produced oxygen that allowed us to live on this planet. It made the planet habitable. These microbes occupy every ecosystem on the planet. They occupy a range of different niches. We are increasingly starting to understand the role of pathogens, especially in the past few years. But there's a range of other microorganisms, bacteria, archaea, fungi, and viruses that actually are able to provide beneficial ecosystem services. But we know very little about the diversity and functional attributes of these microbial communities. So there are a number of reasons why it is quite difficult to understand the complexity of microbial communities. So microbes are numerically abundant, right? A common example is that a teaspoon of soil has more humans than exist on the planet and all biological entities. There are essentially trillions of these microorganisms. The other thing that makes it very difficult, in addition to the numerical abundance, is that these are biological entities. So while we are trying to study microbes, they are continually exchanging genes, losing genes, and transferring genes among each other, right? So that makes it very difficult for us to get a true sense of the relationship between microbial diversity and the functional attributes that we gain from them. The other problem is that every single system that you look at, whether it's a host-associated microbiome, Belinda made reference to the human gut earlier, or whether it is a soil ecosystem, every one of these microbes are disproportionately affected by the environmental conditions which they occupy. So in our group, we've sort of set ourselves a bold mission to understand and try and get an ecologically guided context of the role that microbes play across a variety of different ecosystems. So we do this by applying the veterinary One Health lens framework in order to understand microbial communities. We've got research in our group that focuses on human health. We try and understand, for example, how microbial community actions in your gut may lead to stunted growth. And I'll give one example of this study. We try and understand animal health. In these studies, we try and understand, for example, how antimicrobial resistance is a growing scourge, and try to understand its links with the environment and the potential contribution of environments in terms of shaping and increasing the proliferation of antimicrobial resistance. We also have a range of other studies where we try and understand environmental health. There's a new program in our group where we're trying to understand air quality and how that impacts on the capacity to acquire airborne diseases, but also try to think of air quality not only as something that is chemical in its nature, but potentially as microbes exacerbating the effects of bad air quality to lead to further health problems. So of course, I would argue that microbes underpin the One Health concept, right? You cannot study anything that's related to One Health without understanding the role of microbial communities. So hopefully by the end of this talk, I've convinced a bunch of you to become microbiome scientists and to apply your studies alongside all the beautiful studies that you do on elephants, giraffes, and all sorts of amazing things. So, as I said, microbes underpin the One Health, right? Microbial communities play an important role in climate change. We know, for example, that microbial communities in the oceans are responsible for taking up a large proportion of carbon dioxide that is produced through anthropogenic activities. I'll give an example of one study later on where we're trying to understand how changing environmental conditions might actually impact or exacerbate the effects of climatic changes. So microbial communities, of course, have an important role in infectious disease. I won't talk a lot about this because I'm sure you look like you're old enough to have lived through the COVID pandemic. So microbial communities are also important because the latest research shows how microbes can actually play a role in chronic disease. There's an increasing amount of literature that shows the role that microbes play in, for example, mental health. And what you actually eat could exacerbate or worsen feelings of depression in some individuals. But we don't quite understand the precise mechanisms that microbes use for this. Of course, microbes are important in terms of food production, and I've already talked about their role in pollutants. So there is no single one of the SDGs that is not in some way related to microbial community actions, right? I will not go through each and every one of them, but for example, the consumption of microbial products is quite core to SDG 1. We know that microbes are important in terms of fostering a biotech startup, and so they could be a solution, especially in our country, towards the development of startups that can employ the vast majority of graduates that are currently unemployed. As I've said, I can argue about the role and importance of microbes in every single one of the UN Sustainable Development Goals. So, of course, all of us know now, if I didn't mention the words AI, I would be very unfashionable. But there is a lot of research that's shown the importance of microbial communities and how we can apply machine learning algorithms, whether it's in terms of finding potentially new enzymes or bioproducts, or whether it's in terms of finding potentially novel probiotics that can be used for human health. But the big problem with all of these AI algorithms is that they require biological data, right? And in order to harness the microbiome for public and ecosystem health, we need to build a diverse training set that minimizes bias and prevents misleading or inaccurate system behavior. What do I mean by this? All of these AI algorithms are trained with biological data, which is largely from the global north. There's very few data sets from the African continent, and I don't need to convince all of you that we live in the most biologically diverse continent, and of course it would make sense to include microbial data sets in any kind of AI development tools. So this image shows, it's an image from a perspective we published a couple of years ago. So the yellow and green dots show the current data sets that are stored on important databases where all of us deposit sequence data, right? So what you can obviously see is that when you look at our continent, there is a lack of biological data sets that are linked to metagenomic data. And when you look, most of them will be yellow because that has been largely driven by researchers in the US who have used microbiome data sets primarily from the Hadza populations in Tanzania as a potential comparative tool to understand what early colonization microbiomes looked like. And there is a lack of data sets from the vast parts of the African continent. And this is something that we are working with others to try and close this gap. We need data that speaks to understanding the role of microbes in engineered environments. We need data that focuses on environmental microbiomes, and we also need data that also focuses on human health or animal health in terms of looking at host-associated microbiomes. So with a few others, we ambitiously called for the establishment of the African Microbiome Project, where we would like to sequence 10 million samples in the next 10 years. We specifically would like to sequence samples of clinical and environmental importance. And as I've said, these data sets are important in terms of any kind of development of artificial intelligence models that use microbiome data sets. So we've done this, and we called for this very ambitious project with very little support, using the field of dreams quote that I like very much, that if you build it, they will come. And by they, I'm talking to any of you who've got good checkbooks that would like to support any of our efforts. And so one of the things that we've started to think about is what kind of infrastructure or ecosystem do we need to develop microbiome research on the African continent? We know, for example, that there is a lack of coordinated groups of researchers that study environmental microbes, right? A vast majority of microbiology that's done on the continent focuses on culture-dependent work that tries to elucidate the taxonomy and structure of microbial communities by growing them in the lab. There's very little metagenomic work that's done throughout the continent. And we've worked with a few international societies to try and think about how we can establish coordinated networks to try and work on questions related to the microbiome. We've also started to think about what kind of policy interventions are required to allow us to have the framework to be able to undertake microbiome studies throughout the continent. The other thing that's quite important is because a lot of this depends on us being able to undertake large amounts of genomic sequencing, we try to think about whether it would be possible to establish different genomic hubs throughout the continent in order to support the development of microbiome research on the continent. And as we do this, we've also started to think about exactly how this research should be done. I'm pretty sure most of you here have worked with people working in the conservation space, where most of the intellectual programs are led by researchers in the global north, and African individuals often serve as people who provide the samples, who enable people to come in and actually do the work. But there is very little intellectual involvement within the projects that are done on the continent. The same problem persists in African microbiome research. And we've outlined here in Nature Medicine a framework of how we should collaborative in order to allow for equitable microbiome research. So as I've said, I'll give you 3 examples of how our group has started to close the knowledge deficit related to microbiome studies on the African continent. I'll talk a little bit about a project that's one of the projects that's linked to environmental health, human health, and also animal health. So I'm showing this picture because, I had a little bit of FOMO because everyone has their pretty images. And unfortunately, because we don't grow these microbes in the lab, we don't have anything as photogenic, but please enjoy. This is an image of a research cruise that I went on, where we tried to study microbes in coastal marine ecosystems throughout the continent. This is a program that was supported by the European Union, and it allowed us to understand how the influx of waters from African rivers into the Atlantic Oceans shapes the structure of the Atlantic Ocean and influence biogeochemical recycling in this very important marine ecosystem. So as I said, the first story thinks about, or is related to the role of microbes in climate change and how they may potentially exacerbate the effect of climatic change. So I'll try and explain each of these images. So in this work, which Maibonge Buthelezi carried out, we are quite interested in understanding how microbes that are at the interface of sea ice and sea water, how those ecosystems will change with the increase in temperatures. So we use sea ice, we collected sea ice cores, and also seawater. And we specifically try to understand how one volatile compound, DMSP, it's got a pretty long name which I can't say very well, but it's a sulfur-related compound that is related to the sulfur recycling in marine ecosystems. So the important thing about this DMSP is that it produces DMS, which is a volatile gas that has been shown to contribute towards increasing the greenhouse effect in different ecosystems. So what Maibonge tried to do is he collected samples on a cruise that was sponsored by the Department of Environmental Affairs from the coast of Cape Town to Antarctica. And then we measured the levels of DMSP in each of these samples, right? I'm not sure if you can see this, and the pointer doesn't work, but image B shows the concentrations of DMSP across different samples. You don't need to see each of those images, but what you can see is that the first large point indicates the DMSP that's stored in sea ice. As you can see, the quantities are more than 4-fold higher than those that exist in seawater. Of course, this has implications in terms of what will happen as sea ice starts to thaw and this DMSP gets released into the environment, right? It could have quite profound effects on altering ecosystems. The very pretty phylogenetic tree there shows the diverse assortment of microbial communities, some of them which represent pretty novel lineages that have not been described previously, that play a role in the recycling of DMSP. And we're able to describe several novel genetic mechanisms related to DMSP recycling in the Southern Ocean. So the second story that I'll share with you is work done by Isabella Blumeris, who in her work tried to understand the effect that, try to understand the role that microbes play in the growth and development of children who are born from HIV-positive mothers, but who were on antiretrovirals. So these kids are not HIV positive. But what other studies have shown is that when they look at the growth trajectories of these kids and compare them to peers, they demonstrate stunting, right, compared to peers of the same age. And our hypothesis is that the microbiome plays a role in terms of contributing to the growth stunting of these individuals. And what we tried to do is we took a bunch of stool samples, and I'll rush through this, and what we could actually see is that genetic mechanisms that are linked, for example, to short-chain fatty acid metabolisms are quite diminished in kids that are stunted, born from HIV-positive mothers, compared to peers of the same age. So this work shows that the microbiome plays an important role in terms of contributing to growth stunting in South African individuals. The last study, which I'll quickly share with you, is a project that we did just shortly after COVID in the Limpopo province. And what we tried to do in this work is that, classical water analysis rely on being able to measure fecal coliform counts in order to determine whether water is good enough quality to drink. But the problem with these tests is that they are very good measures, but they don't really tell you about what other problems could be present in the water. For example, they don't give us any kind of indication of the extent of antimicrobial resistance bacteria within these environments. So what Nyasha did in her study is that using knowledge that we have from other studies where we've seen that there is a specific type of virus that is a very good molecular marker or is linked to people that the stools of individuals and also animals. So this marker, the crassphage gene, can actually give you information about the level of antimicrobial resistance in these individuals. So what we thought is that we could probably use this marker as a better indicator of contamination in these waters. And what we're able to show, and the paper is published, so you can also have a look at that, is that the crassphage virus is a much more sensitive indicator of contamination in these waters. When we looked at fecal coliform tests, which we did alongside the analysis that we ran, often when fecal coliform tests indicate that the water is drinkable, it misses the high amounts of antimicrobial resistance genes, viruses, and other organisms that are present within these waters. So what we'd like to do in future is see how we could develop a molecular-based marker for determining contamination in water sources. So in summary, what I've tried to share with you is that microbes are quite important in terms of the role that they play in terms of climate regulation. With Isabella's studies, I've shared that these microbes are also important in terms of human growth and also nutrition. And in the last work that I shared is that understanding viral diversity can be used as an important marker of pollution in water sources. So all of these stories together highlight that microbial communities link every component of the One Health concept, including the environment, human, and animal health. So ignoring microbes means that you're actually missing the foundation of environmental and public health. With that, I'll stop my talk here, and thank you all for listening.
Thank you, Thulani. We have time for some questions, if anyone has questions.
Don't all speak at once. Sorry.
Please give your name and affiliation.
Thank you very much for the presentation. I just have one question on how you bring all this together. You presented very good examples, different studies. So do you have like a One Health indicator? Are you working on like a composite One Health indicator that brings all this together?
I think that will be difficult to do because it really depends on the ecosystem. But for example, in the last study, I've shown if you wanted to look at fecal-related pollution in water, that using viruses might be a better indicator. In other studies, for example, where we've looked at harmful algal blooms in coastal marine environments, in that case, you might need to use changes in community composition related to certain cyanobacteria to understand how that impacts the ecosystem, right? And to use those as probably a better indicator for determining mycotoxin contamination. So in the gut, for example, the type of indicator you'd use in that instance would also be different, right? So I don't have that one magical indicator, but I guess if you are willing to accept microbes as that magical indicator, that's what you would use.
Thank you. Does anyone else have a question? Okay, thank you, Thulani.
Cool. Thank you.
Just a quick housekeeping. A cell phone was found on one of the poster stands. So if anyone is missing a cell phone, please head to the registration desk to collect it. Our next speaker for today is Dorette Pretorius, who will be sharing insights from a comparative study evaluating cheetah survival and reintroduction outcomes in KZN reserves.
Wow, this is not intimidating at all. Good afternoon, ladies and gentlemen. Thank you, Madam Chair, and also thank you to the Oppenheimer Research Conference for allowing me to come and present some of my results for my master's project. So I did my master's through Tshwane University of Technology, and I basically looked at the evaluating survival and reintroduction success of cheetah in 2 northern KwaZulu-Natal reserves. So as we know, cheetahs face various threats, but even though they are classified as vulnerable by the IUCN, the global population is still declining, with only about 7,000 mature individuals left worldwide. Southern Africa holds the largest remaining population, with about 4,500 mature individuals. And it's sad to say, but cheetahs only occur in about 10% of their historical range. In South Africa, only 44% of prime cheetah habitat actually is within formally protected areas, therefore making regional conservation efforts critical. But to mitigate the population declines, reintroduction programs have been widely implemented. So just quickly, the cheetahs in South Africa or Southern Africa are managed as a cheetah meta-population through coordinated translocations. And the core strategy of this is just basically to maintain genetic diversity, to provide demographic stability, and to reduce the risk of local extinction, and also to support reintroductions into historical ranges. In South Africa, a network of more than 67 fenced reserves and more than 460 cheetah form part of this cheetah meta-population program. However, despite national strategies, the reintroduction success is variable, even though there is national strategies. So each reserve has a unique ecology, prey and predator dynamics, making standardized outcomes really difficult. My 2 study areas, northern KwaZulu-Natal, Soshanguve and Mfolozi Park, as outlined in red, and Munyawana Conservancy, as outlined in green. Now, in terms of size, the 2 properties is quite different, but the environmental factors are very similar. Therefore, I could actually compare these 2 reserves in terms of the cheetah reintroductions. Soshanguve and Mfolozi Park is a state-owned and managed reserve. They focus on process-based management, basically conserving multiple species and community survival, although some species are prioritized for their conservation value, and at this stage it's only the black and white rhino and also the African wild dog. Munyawana Conservancy is a communal and privately owned and managed reserve, and they focus on their operational management is basically driven by high-end tourism, and they focus on landscape level biodiversity, with certain priority species, so therefore protecting important catchment areas, threatened, rare, and endemic species. So just quickly, I want you to focus here. In 1966, the first cheetah introduction were done in Sihluwe Park, and up to 2023, there was 113 cheetahs that were reintroduced. And within that same time frame, only one cheetah got translocated from Sihluwe Park. In Munyawana Conservancy, the first cheetah reintroduction was done in 1992, and up to 2023, 45 cheetahs were reintroduced, and in that same time period, 81 cheetahs were translocated from Munyawana Conservancy. So basically, it's the continued introductions from Sihluwe Park, yet no outward translocations. So it seems like Sihluwe Park is acting as a sink population. And this discrepancy raises important management, ethical, and resource allocation concerns. So I basically tried to assess if continued introductions are justifiable, and I basically done that by determining the current, or also to determine if the current introduction strategies align with broader conservation objectives. And I've done this by investigating survival patterns, mortality causes, and also introduction effectiveness. I use Kaplan-Meier estimators, which is a non-parametric step function, which basically records the lifetime data at different points. And also, I developed a novel short-term assessment model to assess these reintroductions. Just very quickly, I'm not going to spend too much time on this, but there's just some figures of the amount of cheetahs that underwent the survival analysis. At the population of the start of the study in Munyawana Conservancy were 15 and Sihluwe Park 14. And at the end of the study, 31 individuals, cheetahs for Sihluwe Park and 28 at Munyawana Conservancy. The total deaths are quite strong in Sihluwe Park at 61 and Munyawana Conservancy 113. Right, so here we have the first set of results for my survival analysis. The graph on the left-hand side compare the two reserves. You see the median survival is basically identical, so that means that both parks lose half of their population in about the same time frame. So that means that the survival challenges is quite equal in both of these reserves. The crude survival percentage is also very similar with 35 and 34%. On the right-hand side, I have done the survival curve for introduced individuals at Sihluwe Park and also at Munyawana Conservancy. Again, no statistical difference in terms of the median survival time, but the survival percentage also quite similar, 45 and 50%. Here I have actually done survival curves for all the females and males combined in the reserves. There's no statistical difference in terms of the median survival time. 65% survival rate for males and 33% survival rate for females. So females fare, in terms of crude survival percentage, they've got a clear survival disadvantage. And here I've actually split it up. I've done Munyawana Conservancy separate and also Sihluwe Park. Again, even though there's no statistical difference for the median survival times, crude survival percentage is actually quite different. And this is what we as managers, reserve managers, really want to look at, because that will influence our management strategies. So here the female cheetah undergone the survivability percentage much lower, regardless of the park. And this is not an isolated issue, but it is a systematic challenge facing cheetah conservation. Here I have compared the survival rates of reintroduced cheetah with those that had previous encounters with leopards, lions, and hyenas, and those that came from reserves without those specific apex predators. Again, no median survival time did not show any statistical difference, but the crude survival percentage was quite significant. So experienced cheetahs, or experienced predator-savvy cheetahs rather, is likely to survive 4 times longer than those without any experience. Here we look at the relationship between age at introduction and also the post-release survival. There is a moderate statistical significant difference at the age of what the cheetahs were introduced at. So the older cheetahs at introduction survived for longer periods than those that were introduced at a much younger age. The R-square evaluate, it's basically indicating to us that only 15% of this variance can be explained by the age at introduction. So somewhere there are other factors that plays a major role in the success post-release. Just quickly, the mortality here, the causes of mortality. I would like to point out that on the right graph, that cream-colored bar, that indicates the unknown mortalities in Soshanguve and Mfolozi Park. That was about 70%. So it's quite high in terms of uncertainty what causing these cheetahs to die. But most natural mortalities occurred from lions and other cheetah. In Munyawana Conservancy, lions killed mainly adult cheetah, of which most were female, and 66% of male cheetahs were also killed by other male cheetahs. And Soshanguve and Mfolozi Park, some cheetah males were killed by lions, but all female cheetahs were killed by lions. And no cheetahs were killed by other cheetahs in Soshanguve and Mfolozi Park. So to assess whether reintroduction programs are working or reintroductions are working, the IUCN has set some criteria that you can use to compare or to analyze your results. But I found that this is mainly designed for a broader translocation efforts, and is also much longer term and not really short term. So specific metrics such as establishing a population of more than 500 is unrealistic for cheetah specifically in any given reserve. Also, the IUCN criteria focus on population level outcomes and not really looking at the individual success. Therefore, I developed a short-term analysis, reintroduction analysis. I called it RACE, Reintroduction, Acclimation, and Cheetah Establishment. This allows us to move beyond population level to assess individual success. And this also integrates ecological and behavioral indicators into a single composite score. So this basically allows us to compare directly the outcomes between reserves, individuals, and across landscapes also. So this model highlights the specific strengths or concerns when we're dealing with introduced individuals, and it also allows managers to actually have real-time adaptive intervention, and that also standardizes monitoring because you have to take into account certain factors. This is just an excerpt of the model that I have developed. Each indicator is basically scored from 1 being unsuccessful outcome up to 5, which is the ideal outcome, and you basically go out into the field, you collect your data, and you come back and you then score these individual indicators. You then calculate the total score, and then divide it by 40, which is the maximum amount of points you can receive, and then it's basically expressed as a percentage, which I call it the score percentage. And here you would take that score percentage and you would compare it to the score percentage range to obtain a success score level. So all the cheetahs, I applied this model to all the reintroduced cheetahs at both the reserves. And basically I found that the average reintroduction evaluation scores are very similar. Both reserves are scored on level 3. Which basically means that there's generally positive outcomes, although there is continued support and monitoring needed in both these reserves. Right, so from that also, the evaluation or the short-term introduction success depends then on previous predator experience and also successful hunting. Longer term depends on cheetah population size, whether there would be enough partners for the female to select a breeding partner, and then also intraspecific male competition. So what we know now is that the overall survival is similar, although it is driven by different factors. The key drivers for success are individual traits, not necessarily population traits, and that Soshanguve and Mfolozi Park is not necessarily a lost cause. Population was below minimum viable size to actually have good production. And then Munyawana Conservancy is a proven source, because there we can actually get export quality cheetahs. So my recommendations then after all of this is that when we do translocations, we need to look at prioritizing to release cheetah coalitions rather than single males, select individuals with prior leopard lion hyena experience where possible, and be cautious when we're introducing younger individuals, especially at high-risk reserves. In terms of monitoring, we need to have mandated improvement monitoring at Lwandile Park, but there's also lots of other reserves that can't afford necessarily to have monitoring programs. And in terms of management, if we want to increase the cheetah survival, we have to lower lion densities. And if we want to continue, well, we can continue with the reintroductions such as Lwandile Park if all the above criteria has been taken into account. Yes, and then we have to celebrate and support Munyawana Conservancy's efforts in creating these translocating cheetahs. Right, and then I would just like to thank all my supervisors, Tshwane University of Technology, the 2 reserves, and those guys who had the boots on the ground, and my family and friends for listening to all my ramblings about cheetah reintroductions. Thank you so much.
Thank you, Dorette. We have time for one very quick question, if anyone has one.
Please don't ask me how many spots a cheetah have.
We have one over there. Please, can you just state your name and affiliation when you ask?
It's Declan Hoffman here. On your race score, maybe I misinterpreted it, but 0% you still said is regarded as failing. So if 0% is still regarded as failing, what is a failure?
So if you look at all those individual scores that have been allocated, if most of them have scored either 1 or, basically 1. So that model, I think at this stage, looking back at it now, I need to adjust it a little bit to actually account for smaller how can I say, indices, so that there's a clear, not because there's only 4 criteria at the moment, I would like to actually have it more precise. But it's basically failing on all of those factors like settlement, hunting, breeding,
okay.
And also not being able to defend themselves against other predators.
Does the managers like me like a nice line where we can say, okay, it's time to make a decision? And
Yeah.
if everything just says failing the whole time. And then, sorry, one other question, because this is something that I'm actually involved with at the moment with the introduction. What is the lion density? Can you say what the lion density is that favors cheetah female survival?
Well, in those specific reserves, it was quite different because of the large size of Lwandile Park and Munyawana Conservancy. But because of the smaller size of Munyawana Conservancy, the lion density were much larger. So if I can remember correctly, it was about, at one stage they had more than 100 lions. If I can remember correctly, I think it was 1 lion for every 25 hectares.
Thank you.
Thanks, Dorette.
Thank you.
Our next speaker is Gabriella Kietzka, who will be sharing her study evaluating the success of conservation corridors for dragonflies within plantation landscapes.
I love that I even have a soundtrack when I walk on. Good afternoon, everyone. My name is Gabby, and I'm from Stellenbosch University. I'm currently in the biomaths department, where I'm busy with a postdoc, and I'm going to be presenting a paper that we currently have being reviewed based on using zeta diversity, which is a novel framework used to assess biodiversity patterns and looking at conservation corridors in plantation forestry in KwaZulu-Natal, and using dragonflies as indicators of corridor success. So as I'm sure most of us here are aware that we are in trouble. We are currently facing the 6th mass extinction, And this is caused by anthropogenic causes, such as land use, agriculture, including forestry. And with our population ever growing, the more people we have, the more resources we need. And there's a need for immediate conservation action in a way that we can try and meet that balance between human needs and ecosystem preservation. So the focus of our paper was on plantation forestry in KZN, and this is one of the biggest, well, in KZN, it's one of the main causes of landscape degradation. Almost 40% of the natural landscape has been transformed into forestry plantations. But I mean, we all need paper, we all need toilet paper, so we have to find a way to reach a balance where we can somehow try and conserve the natural ecosystems that we still have. So one of the major problems with plantation forestry in this area is that it's conversion of natural grasslands into these monocultures of exotic alien trees, mostly comprising pine and eucalyptus species. So it's not just a landscape change, it's also a structural change. You go from these open grasslands with lots of sunshine to these really, I mean, I've done a lot of work in these plantations and there's nothing under them. I mean, eucalyptus species specifically, they release some kind of chemical that nothing grows under them, and it's a very barren area. So one way that some of the forestry plantations, so the places we worked in, we were funded by Mondi, and they've tried to implement a conservation strategy that seems to be working. It's been for a good couple of years now, and these are called conservation corridors. So what a conservation corridor is, it's basically trying to keep either remaining strips of natural grassland between these plantations, or to regenerate areas that were once planted into a natural state. So that's an example of one of the sites that we worked on. So my main interest has always been in freshwater ecosystems. And a lot of these pine plantations, these corridors, have fresh water bodies in them because of the regulations that forestry companies have that they're not allowed to plant. I think it's 30 meters at the moment, close to fresh water sources because of how these exotic trees take up a lot of the water and mess up the water sources. So it kind of makes sense that if they're going to have these natural corridors, that they include freshwater systems in them. So the main purpose of these is to reduce habitat loss, so by creating additional habitat for the natural grassland species, and also reduce fragmentation, because in these plantations you still have some natural protected areas, but they're kind of patchy, so it reduces the ability of species to move across them. So corridors help facilitate that. And this is just some of the reasons why, or the purposes behind implementing corridors. So as I mentioned, habitat connectivity helps create gene flow, especially with species that you want to reach these genetic bottlenecks. So whether they're using these corridors as additional habitat or just for migration between natural areas, either way it's helping with gene flow. And then obviously, if you want to look at the benefits to us, you look at how ecosystem services are preserved by conserving these natural grasslands. And specifically with my interests, fresh water, because we all need water. So this is just a basic picture of what they're trying, or what they have produced using these corridors. So these corridors link to either already existing protected areas, or just like natural patches of area. And if you have multiple corridors connected to each other and these protected areas, it produces what they call a landscape ecological network. So it's not just one line from one protected area to another, it's a whole network of them that together helps maintain biodiversity and conserve our natural grasslands in the area. So that's a really nice picture of what an ecological network kind of looks like. So where I'm standing is on I think it was a nature reserve, and that corridor between is obviously an implemented corridor. And also why I like this picture is that you can see in that corridor, there is also a road. So it's not that it's just completely natural. It can comprise a multitude of things, but the idea is to try and get it as close to the natural grassland state as possible. And there's a dam, which is not natural either, but it's still a fresh water source. And if you look behind it, that's another natural area with a bit of natural forest. So in terms of looking at the success of these corridors, there's been a lot of research going on for multiple years. And a lot of this research has been based on looking at terrestrial examples. So there's been work done on grasshoppers, butterflies, dung beetles. There's also been some plant work, but why not look at the freshwater ecosystems too? So I've been studying dragonflies since, my undergraduate years. So what they found with these corridors and looking at the insect species as indicators is that they are working. And what we can determine from these different studies is that the wider the corridor, the better. And the more heterogeneity you have within that system, the more species needs you can accommodate, whether it's butterflies, whether it's dung beetles. So the main highlighting point of all of these different studies, I think there's about 20 of them, is that we need to conserve heterogeneity in all its forms within these corridors. And also try to reduce edge effects. So if the corridors are too narrow, they still face the issues that they do when close to those pine plantations or eucalyptus plantations. So I can speak about dragonflies and damselflies forever. They are my passion, and they are highly sensitive to environmental changes, which is why we use them as indicators. So in terms of them being used as indicators, they can be used in a variety of ways. They can be used to tell how healthy an ecosystem is, or in terms of this project, how well a conservation effort is working. But other work that we've done, we've also used them as umbrella species, which means by protecting the ecosystem or the habitat that they are naturally found in, what percentage of other species are you also protecting? And they've found to be highly beneficial in terms of protecting other freshwater invertebrates. They're also highly efficient at conserving, in terms of umbrella species, terrestrial invertebrates, and also wetland plants, because they have life cycles that depend on both the terrestrial and the aquatic environment, so they kind of can help determine whether both of them are equally functioning well. And then over and above that, we use males when we do sampling because they are highly territorial of their waterside territories. They are massive, they're easy to sample, and if you happen to miss one on a day, you can go back the next day and it's still going to be in that environment. And so they're easy to identify to species level and low cost and easy to teach other people how to use them. But over and above that, I can go on forever about why I love them. So a recent paper came out about how everyone thinks butterflies are the furthest traveling insect. They're actually not. It's one dragonfly called the globetrotter. An even better paper that recently came out is so when females are ready to mate, they come to the waterside territory to find a male. And if the male harasses them too much, the females drop dead. They play dead. They drop from the sky and lie on their backs so that they don't have to mate. But I can go on and on about them forever. So what made this study different to other studies that have been looking at corridors is that we applied the Zeta diversity framework, which I'll get into in a bit. So up until now, we've mainly been focusing on like alpha and beta diversity metrics, such as species richness or like species indices. And I mean, the dragonflies even have their own specific dragonfly biotic index. But the problem with these pairwise comparisons is that they don't consider the roles of rare versus common species, whereas the Zeta diversity framework does. Don't ask me about the stats behind it. Ooh, that was fast. I'm normally way too quick. Okay. So the Zeta diversity framework enables us to compare species turnover between any number of sites. So if you're looking at species between any two given sites in an area, that is if a species is shared between only two sites between multiple areas, that is considered a rare species. But if you compare 5 sites and the same species is found between any 5 combinations of sites, that is considering the more common species. And Zeta also considers the effect of distance on sites, which is an important variable when looking at these kind of effects. So that's just a quick depiction of what I was trying to show. So if a species is shared between the third order zeta, it would be a more common species shared between any 3 combination of sites compared to first order, which is alpha, and second order, which is beta diversity. So we wanted to assess diversity in plantations, and we looked at corridors compared to natural protected areas using the zeta diversity framework, but also looking at richness and the influence of environmental variables on these metrics. So we had 104 river sites within corridors and within natural areas, 55 natural, 49 in corridors. And each sampling session comprised 2 observers looking at dragonflies on clear, windless, hot, sunny days for an hour per site. And as I mentioned, only looking at adult male dragonflies. So we collected a wide range of variables, but based on previous studies and the questions that we wanted to ask, we selected these 5 to look at because invasive species as well as the land use type it looks at the disturbance side of things, whereas dissolved oxygen and water temperature is focused on the river dynamics, and shade cover and dissolved oxygen is focused on, shade cover is more like the terrestrial side of things, which all interplay with each other. Oh, flip. Okay, let's speed this up. So we used zeta, diversity, and species richness estimates, and considered how those variables affected them. Now, the most important thing is that we wanted to see whether these corridors were behaving in the same way as the natural areas to determine whether they are actually being successful in terms of species conservation and ecosystem preservation. Okay, so I'll go through this quickly. So we recorded 37 species, 20 Anisoptera, which are your typical dragonflies, and 17 Zygoptera, which are the more delicate, sensitive damselflies. So our unique findings, I'm not going to go into details, but in the corridors there were 4 unique species, and one of these was actually a highly rare and threatened species. Whether it was using the corridors just for movement purposes or actually as a habitat, we don't know, but it was a really cool finding, especially for the plantation company that we were funded by. And natural areas only had 1 unique species. So in terms of species richness drivers, only land use and shade came out as important drivers of species richness in these areas. So corridors actually had higher species richness than natural, which you think, okay, that's a bit odd, but again, it might be because they're using these areas for transport, and also might be because there's more generalist species that are inhabiting that areas, but I'm not going to speculate, it's just thoughts. And then shade cover also influenced species richness and reduced it as shade increased, species decreased, which is to be expected because they thermoregulate and depend a lot on sunlit areas. So in terms of the zeta diversity framework, so if you look at the natural and corridor sites, so corridors had more shared species as you go down than the natural areas did, but it wasn't significant, so it's not really something to focus too much on, but that these corridors are not performing in a way worse than the natural areas are. So then looking at zeta 2, when we looked at those environmental variables, those are the more rare species because they only shared between two areas. And those are the variables that affected them. So increases in those variables typically reduce the similarity between sites. And you'd expect rare species to be sensitive to more variables. Okay, I'm going quick. And in terms of the common species, they had fewer variables, but what's important here is that neither, for both of these, neither land use nor invasive species came out as predictors, which means that these corridors are possibly functioning in a way that is not different to that of natural areas and controlling the invasive plants. So conservation significance, corridors prove themselves as a good way to conserve dragonfly diversity, as well as previous studies and diversity in general, and the broad application of zeta diversity ultimately is showing us that this should be applied to many other biodiversity studies, and corridors potentially be used in other land transformation types too. Whew. No time for questions, sorry.
Thank you, Gabby. Yes, unfortunately, we don't have time for questions. So if you have questions for Gabby, please catch her over afternoon tea.
Well planned.
Our next speaker in this session is Lwandile Ngozi, who will be speaking to us on biological indicators and responses to changing climates and landscape use in freshwater ecosystems.
Good evening, everyone. As introduced, my name is Lwandile Ngozi, and today I'll be talking to you about my PhD project, which is looking at water quality and biological responses to land use and climate changes in the KwaZulu-Natal rivers. The project is supervised by Professor Colin Downs and Dr. Matthew Bennett. Freshwater ecosystems have been pivotal to supporting human livelihood for as long as human history exists. As such, these systems have been greatly impacted by human activities. To this day and age, Sustainable Development Goal number 6 and 14 of the United Nations, which is clean water and sanitation and life below water, are still a major concern. The climate and land use changes are seeing some of the major rivers deteriorate beyond acceptable ecological conditions, and in the process limiting their ability to support biodiversity, provide water resources and related ecosystem services. Climate-induced extremes such as floods and droughts exacerbate the impact of pollution in stream barriers, as well as available aquatic habitats, thereby impacting the overall ecological functioning of aquatic ecosystems. The lack of enforcement of the already existing legislative framework and the lack of maintenance of the water infrastructure exacerbates water demands in the growing populations, as well as changing climates. So our study aimed to understand the impacts of climate change and land uses on the KwaZulu-Natal catchments. We wanted to understand the ecological responses of selected sites to climate change above and below rainfall events. Our focus was mainly on benthic diatoms and microinvertebrates as biological indicators of water quality. So we objectively assessed microinvertebrate communities at selected sites in the KwaZulu-Natal catchments. And we also monitored water quality and chemistry across these catchments. So these are the 52 sites across the 3 catchments, which is the Umkomaz, Umngeni, Uthukela, and all the way to Upongolo and Ngwavuma. So to collect physical chemical data such as your pH, temperature, we used a probe with multimeters, and we submerged it in situ to get those results within the sites. And we also collected water samples with 2-liter milk bottles, which were then sent to Umngeni, Uthukela laboratories for chemical analysis. So for E. coli monitoring, we used 500 ml bacterial bottles that were also provided by the Umngeni Uthukela lab. To collect benthic diatoms, we would then scrub rocks with a toothbrush and then preserve the residue in ethanol, which we have sent to laboratories for analysis. And then we employed the standard size protocol, where we use a 30 by 30 centimeter mesh net of 1,000 microns, and then we sampled boulders and stones for 2 minutes, and then stones out of currents for 1 minute, vegetation in stream for 1 square meter, and then 2 linear meters for marginal vegetations. And then for gravel, sand, and mud, we spend 1 minute sampling those. So the water quality results were within acceptable levels by the South African water quality guidelines, except a few sites that exceeded those thresholds. And for E. coli, we compared our E. coli results from high flow of 2024 and the low flow of 2024. And we could see a clear difference between these. During the high flow, a lot of sites had higher than normal E. coli levels, and they exceeded thresholds of potential concern. However, some of the sites during the low flow, the E. coli levels lowered, so which could be attributed to high flows, washing everything, even the open sewers, into the stream and contributing to the overall levels of E. coli. And then for the SAS score between the 3 seasons, including the high flow of 2025, we could see clear differences in our SAS score. Mostly the sites upstream with cleaner waters just from the mountain springs showed to have high sensitive taxa, improved water quality, and overall improved SAS scores. However, as we sample down the residential areas and towards the estuaries, some of the sites had very poor SAS scores. The average score per taxon is basically the contribution of each taxa into the overall score of that site. So most of the sites, including the upstream, were within acceptable ASPCT scores, and they indicated that some of the taxa found upstream and downstream contribute to these scores. So overall, land use changes impact river and ecological functioning of those. And with these results, the analysis is still ongoing, but what we want to advocate is stakeholder engagement into developing resilient policies around water management, as well as protection of our lovely ecological freshwater ecosystem. Thank you.
Thank you, Lwandile. We have time for some questions, if anyone has questions for Lwandile. Thanks, Lwandile. The last talk for this session is by Lindokuhle Dlamini, who will be sharing how fire exclusion and grassland degradation impact soil carbon dynamics in South Africa's Afromontane grasslands.
Good afternoon, ladies and gentlemen. As introduced, my name is Lindokuhle Dlamini, and I come from Makhanda, so Makhanda forever. So I come from Rhodes University, but the work that I'm presenting today is actually the work I did during my PhD. You'll see there is a SAON logo, so that's because I was funded by SAON, and it was a joint PhD program between the University of Free State and the University of Burgundy in France. So let's go to the Drakensberg. So next is not working. Okay, so the Drakensberg, if you've never been there, it's actually a 5-hour drive from here. So please do consider to go there. It's breathtaking. So these Afromontane grasslands are found in a temperate environment, and earlier speakers did speak about this. They are in a high altitude region that have high summer rainfall. They have high plant diversity and high endemism, more than 2,500 flowering plant species, of which about 22% are actually very endemic in that region. They're also provide various ecosystem services, which include water delivery and water storage. They are considered a strategic water source area, which means they provide disproportionately more water than the area they cover. They're actually also very important for soil carbon storage. They store the largest carbon stocks in the country, and they can also sequester more carbon. Okay. Sorry? I want to go back. Okay, great. But there's a problem. The problem is these grasslands are found in a climatic envelope that can support trees. While they are ancient grasslands, without fire, they change into a forest-type system. And the impact of this change, largely on soil organic carbon, is not fully understood. So, soil organic carbon have gained interest over the years. And there are several reasons for this, but I'm going to give you 3 reasons. The first one is we know that soils are the largest reservoir of terrestrial carbon. They store 4 times the amount of carbon compared to above ground vegetation, and 3 times the amount of carbon compared to the carbon that is in the atmosphere. But the second one is we are worried about climate change. That if soil stores so much carbon and they also release carbon into the atmosphere through the process called soil respiration, and this is very sensitive to things like global temperatures and land use change. This might start to create this positive feedback loop where soils will start to release more carbon into the atmosphere. But the last one is the hope. The hope that if we can actually improve our soils through effective land management and restoration, we can sequester more carbon and store it into the soil. But again, the hope does come with its own problems. There are simple global push solutions to say, let's plant trees in these open grassy systems, especially those that can actually support trees. So you can see that there is a commitment to plant trees in some of these African systems. So this brings me to my questions. I had only 2 questions. I'm trying to present 2 papers in one presentation. I hope I succeed. So the first one is, does fire exclusion, so when you exclude fire and you allow a system to change into a forest-type system, does that lead to more carbon being stored and less carbon released to the atmosphere? The second one is, what happens when you deliberately plant trees in these areas that are adapted to fires, that have natural fires? So the study was done in Drakensberg, in Cathedral Peak Research Catchment. These catchments were started in the 1940s. They were started to answer hydrological questions, something that I don't know much about. But there was a belief back then that if you can plant trees there, they'll bring more rain and increase the water that we get through evapotranspiration and processes that I don't fully understand. But the answer was no, and some of the policies in South Africa, actually, the data came from this research. So we're actually now using these catchments to answer different type of questions relating to trees, but on climate change now. So there are 3 catchments that I work on. The first one is catchment 9, which is a catchment where we've excluded fire. We've excluded fire, and the catchment naturally change into a forest-type system. But because excluding fire in these catchments is actually on its own a disturbance, it's actually very hard to exclude fire. Around the catchment, there is still a grassland that is maintained through accidental fire that happens every 2 to 5 years, allowing us to compare between the 2. Then we have a natural grassland catchment where this catchment is bent every 2nd year, and it received biannual fires. This is supposed to play, okay. It received biannual fire that are controlled, and we're trying to mimic nature to keep it as a grassland. There's many instrumentation here that say on deployed covariance system, and many measurements are happening in this catchment. The third catchment is a catchment that we actually deliberately planted trees. So they planted pinus trees. After about 20 years of planting these trees, there was a wildfire that completely burnt all of them. So the catchment is now degraded. It looks like that now. It's dominated by a bracken fen, and there is massive soil erosion. After the rainfall, the weir fills up with sediment. So, for the first question on fire exclusion, so what I did was I collected soil samples at different depth increments down to 1 meter and then analyzed the soils. So to look at what did we find, this is moving slower than me. Okay. So to look at what did we find, we actually found that contrary to what you might believe, the grassland actually stored more carbon in the topsoil compared to the adjacent forest. So even though when you look at the whole profile, there is no differences between the two. So when we look at the isotopic carbon isotope signatures, generally the grasses are C4 in that system, and the trees are C3. So you can look at the signature and look at where does the carbon come from. So if you do a simple equation called isotopic mixing equation, you can see that more than 40% of the carbon in the subsoil actually comes from the grasses. It's the old grassy system from that legacy effect when the system was changed. It's the carbon from the grasses, not from the trees. So we also looked at how much carbon is released to the atmosphere, and for this I had to go to the field every month for 1 week for 3 years. So my PhD was very fun. So I was collecting gas samples every month for that 3 years and measuring some other auxiliary measurements. So what did we find? So as you can see in that graph in the top, you see that in the first year after the fire, which is 2019, there was no differences between a grassland and a forest. This is because there was that accidental fire. So the grassland was burnt, so there was high soil temperature, and also it was actually bare. So fire have been known to trigger microbial activity, and it releases more CO2 to the atmosphere. But as the grassland rests, the grassland releases less compared to the forest. Overall, the forest was actually releasing more CO2 to the atmosphere. When we do a simple calculation, which is the Q10, looking at the sensitivity of the two systems to changes in soil temperature, we saw that the forest, afforested area, actually was more sensitive, double the sensitivity compared to the grassland. So let's go to the second question. It looks like I'm going to finish. So the second question is looking at in a catchment that is burnt every 2 years, we use it as a control, and then we have another catchment that was planted with pinus trees, and there was a wildfire. So because these are two catchments that we are comparing, we look at the top, upper slope, middle, and bottom to look at soil movement also. So what did we find? We found that in the grassland, which is a natural grassland, there was more carbon in the upper slope. But when you look at the bottom slope of the degraded catchment, you see that there is double the amount of carbon in the depositional zone. This is normal in soil sciences. Carbon moves with the soil. As the soil moves from the top to the bottom, it will carry carbon with it. But the interesting results are actually what is coming next. When you look at the reactivity of this carbon, looking at active carbon, so the easily degradable carbon, the less stable carbon, you see that in this bottom slope in the depositional zone, most of this carbon is not the stable kind of carbon. So it will easily be degraded and decomposed by the microbes and released into the atmosphere. We also looked at microbial activity. There was someone speaking about microbes, so I did look at microbes. So the microbes also, we see the similar result. In the depositional zone, there is high microbial activity. What does this mean? It means that this is susceptible to be released to the atmosphere, and most of the carbon is actually being released to the atmosphere. It's not being stored in that system. Okay, let's sum everything together. The first question was, does fire exclusion, allowing a system to change into a forest-type system, does it lead to more carbon being stored and less carbon released to the atmosphere? The answer is no. We actually saw that it reduces soil organic carbon storage, changing the system from a fine root system with higher turnover to a litter system that takes longer to be incorporated into the system. And because of fires, some of this litter can actually be burnt before it's incorporated into the soil. It also releases more CO2 to the atmosphere and have higher temperature sensitivity. So this means that actually in Afromontane grasslands, afforestation is not really a good idea. It's not a good climate change strategy in these grasslands. And when we look at the consequences of planting trees, the likelihood they will burn, and that would lead to accelerated soil erosion, which then this study is cautioning against this large-scale tree planting because of the impact on the carbon and impact on soil health. Just like Professor William Bond, who presented here before, have said, plant the right trees in the right location for the right reason. Thank you.
Thank you, Lindo. We have time for a couple of questions. If anyone has questions, we have one here in front.
Thank you so much, Dlamini. Your gospel is home. I feel like this has been a very good day for me because this has been an area. In Kenya, we have even a national holiday for planting trees where none existed. So sometimes planting it in grasslands where it's just an old grassland. But I think the gospel is home. But my question to you is, how do you think this message can be taken to the right people, like the government and policy makers, in such a way that they don't just have to be putting much effort into planting trees, thinking that trees are actually doing better than grasslands? Because it's a gospel that we spread, but which is the best way you think we can send the gospel to them too?
The answer is I don't know. But what I've done, I'll tell you what I've done. I was at Seyon, so after your studies, you write policy brief and you send it to government. So they either listen or they don't listen. And I'm hoping that through presenting in some of these conferences and sharing this knowledge, I also created simple, easy diagrams showing what we found and what it means. We've also written a paper written by all the researchers that are working in mountainous region, looking at the story, the lies, and what we can do to actually protect the carbon in the mountain. We are trying, but I'm not sure if they would listen.
Thank you so much for a great talk and really good data. I wanted to ask you, did you consider the above-ground carbon components? So I think your results are really clear on the soil carbon, but I think the question is, if you were to advocate for replacing the plantations with natural grassland, would the increase in soil carbon compensate for the loss of above-ground carbon, assuming that fire suppression continued and kept that plantation as a stock, if you understand what I'm saying?
Yes, thank you for the question. It's actually a question I've been getting every time I'm presenting this work. The answer is we did not look at it, and I'm hoping that I'll get time to actually look and quantify under Lukicidia. But one of the other problems is we're not looking at carbon only, we're looking at water, we're looking at, let me get this right, albedo. So I always pronounce it wrong. But we've seen that it actually has a problem on the water, so it has higher evapotranspiration and lower albedo. So it's actually a problem. It would be hard to advocate for it to suppress fire. But the other reason why we did not look at above-ground biomass is because the system generally bends. So those trees, in the next few years, they would likely burn because it's becoming really difficult to control fires in that region, like you saw with that pinus area that was planted.
Okay, one more question.
Hi, that's really interesting. I'm Dave Gaynor from the MRI. That's really interesting results. I just wonder, I mean, you planted pine, okay? So how does this apply when you actually plant, when they're indigenous plants in that area? Do you have any idea of the difference that would make?
Thank you for the, yes.
Suppression of fires would lead into slight increase of natural forest as well, rather than this burning every 2 years, for context. Yeah.
Yes, so that the first question was looking at allowing indigenous trees to come in. So it's, even though we did not plant indigenous trees, but the indigenous trees came in, changing a system into a forest type. While it's dominated by Leucaedia, but it's transitioning into an Afromontane forest. And that's where we saw that actually the grassland is storing more. And under the forest, actually most of the carbon still comes from the grassland. Not sure if I'm answering the question.
Yeah, now you cleared it up. I had your experimental design wrong. Yeah.
Thank you.
Thank you to all the speakers. Oh, sorry.
Yeah, I'm very proud of the research that you did and what I've been seeing since I came here. But I've got a question. Is there a way where these research results can be used to produce a documentary which can be aired on a national television so that we educate our young people on the dangers of such activities? Because we've seen a lot of burning of our grass and our forests, and people don't understand why they shouldn't do that. So the researches that we are doing, is there a link between yourself and the Ministry of Information and Publicity here in South Africa to do a documentary so that at least the information can go to the people and the people can learn and understand the dangers of fire? I thank you.
Thank you for the question. Maybe the answer is no, I don't have any relationship with people who can produce documentaries, but I know the organizers for this conference actually have a working good relationship with the media, and I think maybe they can come up with the plan.
Thank you, Lindo, and thank you to all the speakers in this session. Just before we close, I'd like to highlight one of the amazing books that we have, the many amazing books we have on display outside. This is a gardener's Bible, as Duncan calls it, of South African indigenous garden plants. And one of the authors, Elsa Puli, is actually in attendance, so if you grab one, maybe she'll kindly sign it for you. Thank you to all the speakers in this session, and we're going to break for our afternoon tea. Please can everyone be back at 4 p.m.